Nice work. Okay, so tonight we are lucky enough to have Lincoln Besk with us. He will be presenting on his work, Barcoding the Oregon and British Columbia bees. Lincoln is a pollinator taxonomist for the Oregon Bee Atlas and the Master Melitologist program. He has studied the biodiversity of bees and wasps around the world with a focus on the Northwest and the Great Plains of North America. So take it away, Lincoln.
Well, thanks for that introduction, Kate. Good evening, everyone. I'm happy to get an invitation to present here.
Excuse me, Lincoln. I wanted to say one thing. Please keep questions to the end unless you do not understand something that Lincoln is presenting. Then go ahead and put it in the chat and I will bring it up. But otherwise, questions at the end would be preferred. So thank you. Go ahead, Lincoln. Sorry about that.
Oh, it's okay. Yeah, and I'll just motor along here. Okay. Of course, you guys just had your business, and so we have a little bit of business too. We have a new Oregon Pollinator Paradise license plate coming soon. The website is up, and we will be taking orders soon, but not quite yet. So move to Oregon if you have to in order to get this plate. And then another website that's come online recently is Elaine Sedgman's beestoriespublishing.ca. So she's published 2 books for children and other bee lovers, one about Bob, the blue orchard mason bee, and one about Andrina, the mining bee. She's doing lots and lots of presentations. So you can get in contact with her through her website and order her books. So this presentation will have a lot of technical content. For some of you, it'll be a bit more than you're used to. Many of you will be fine. Leave your questions in the chat and we'll deal with them mostly at the end. And I'll do my best at science communication. So I will introduce DNA barcoding and its application, how it can help us with bee taxonomy, the process of barcoding and the products that we get out of it. I'll introduce all of these acronyms. I'll mention what we're doing with all of your collections and your donations that some people have generously given us for DNA barcoding. I'll talk about some of the things we found, and then also the fuss or the controversies and what's next. So within the cell, there are 2 types of DNA. One is nuclear DNA found in the nucleus, and that is the largest part of the DNA that we have, or animals. And then we also have mitochondria, which is what produces energy for the cell. And mitochondria have their own DNA, but they have much less of it than the nucleus. DNA barcoding uses a small section of the mitochondrial DNA. And so you can see here that the bee nuclear genome, the big part of the DNA, is 250 million base pairs, which is about 8% of the human genome in size. So the honeybee genome is much smaller than the human genome. And the mitochondrial genome is even much, much smaller than that. And so the mitochondrial genome is only about 0.006% the size of the bee's nuclear genome. So that mitochondrial genome is really small compared to the nuclear genome. And then within the mitochondrial genome, we only look at 658 base pairs, which is about 4% of it. The mitochondrial genome is a little circle of DNA, as you can see here. And we're just looking at this one little section in here that represents about 4% of it. We look at that section for a few different reasons. We look at the mitochondrial genome because it's highly conserved. Nuclear genes change really quickly and have a lot of variation. Think about genes in the nucleus that might control for hair color or eye color. They're really variable and they evolve really quickly. So those genes aren't good for trying to tell the difference between species. However, if you have really conserved DNA that doesn't have a lot of variation, and doesn't change over time very quickly, it's a lot more practical to use that DNA to compare between species. So there's an advantage there. The benefit to this and to DNA barcoding is that it helps us address this problem, me and the taxonomic bottleneck. But really, it's not just me, it's the fact that we have incomplete, out-of-date, non-existent, or just a lack of available identification resources. We don't have keys for many of these bee groups. We also have limitations in our morphological assessments. So we have many bees and many species, they all look the same. Think about all those dialectists, they all look the same. We can't tell them apart under a microscope. And so potentially quantitative data from DNA could help us. We also have many undescribed species, and many of these are within unrevised genera and subgenera. So whole groups of bees that haven't been studied in potentially 80 or more years. And so they're not well organized, species aren't well known. In addition to that, most places lack any reference material at all. I've worked in 3 primary regions. I worked in British Columbia. There was no reference collection. The Provincial Museum wasn't even curated at the generic level 15 years ago. So when I worked in Alberta, our collection had almost no identified bees in it. And then when I came to Oregon, the Oregon State Arthropod collection of bees is really nice, but most of their good stuff has been out on loan for 40 years. And what's left hasn't been properly, or actively, I should say, actively curated in a long time. And so we're lacking reference material, reliable reference material for the hundreds of species that occur in our area. And finally, because bees are sexually dimorphic, the males and the females of the species look different, we don't have a way to associate them without some other method, in this case, using genetic data. So our process consists of selecting specimens, removing the left side midleg, which is the DNA sample. We then image each specimen and we compile a ton of data sheets. And this is what that looks like. So this is one of our typical collection boxes that I've turned into a grid with 8 rows and 12 columns. So that leaves 96 spaces. And you see that it's populated with 95 bees, with the bottom right corner being empty. This box corresponds to this 96 well plate. So 8 rows, 12 columns, 96 wells, with the bottom right well, the red one, being the control. And so what we do is we take a leg off each one of those bees and we put it in its corresponding well in the plate. And then this plate will hold 95 DNA samples from 95 bee specimens. Along with those bee legs, we take an image of each specimen. This is half of the images for one of those plates. And so you can imagine, thanks to Cody Feuerborn and Phineas McDermott, we've already produced like 1,000 images. And at this rate, it would take a few minutes to show you them all. You're only going to see like 100. Stelis, Coleoxys, who's next? Hoplites, and so on. Lots and lots and lots. So we've already been really, really productive in getting our program underway. So along with those images and with those bee legs, we produce all these data sheets for all the special labels that go on the bees, data sheets for the taxonomy for each bee, for the biology of each bee, for the spatial temporal data and the collectors of each specimen. So here you can see that we have barcoded specimens collected by Julie, Gretchen, Linda, Lori, Stephanie, Steve, Gretchen, Mark, Robert, Pete, Judith, and so on. All of these contributing Oregon Bee Atlas members. And then finally, because we have all this data and all these images and these trays full of bee legs, we associate all that data with that well on the plate that has the DNA sample in it. And so there's a lot of data management that goes into each one of these plates. Once the plate is populated with bee legs, I wrap it up in duct tape, and I ship it to Canada. So we send all of our material to the University of Guelph, which has a world-class facility. They have this Center for Biodiversity Genomics, within which is their DNA sequencing facilities, the Canadian Center for DNA Barcoding. So at the University of Guelph, there's this big genomics center. Within that is the Canadian Center for DNA Barcoding. Those are the people that receive our plates and sequence our bee legs. And then also within that center is the Barcode of Life data system, which is a online software platform that I'll introduce you to. So in September, I actually visited the University of Guelph when I was back in Canada. And you can tell that equipment is really high tech when it has these blue glowing lights. That's how you know. Expensive, high tech. They always have these blue glowing lights. And so, of course, this is a huge multi-billion-dollar facility. And they have all the most high tech equipment. And because of, this equipment has increased in its capacity over the last 15 years, they actually have all the different generations of the equipment, much of it still in working order. So that's kind of the basic introduction. What I want to show you is this Barcode of Life data system. So if you go to boldsystems.org, you too can create an account. It's free, and they have tons of public DNA sequence data, including sequence data for bees within this website. And so I'll give you a tour and a tutorial right now of how this works and what's in there and all the cool stuff you can do with it, whether you have sent them your own bee legs or not. So this is the main console. And from here, you can have a sense of the data sets you've created. And we're going to just jump in and take a look at this project right here called ORBee, which is Barcoding the Bees of Oregon. So the landing page for our project, the Barcoding the Bees of Oregon project, has all these summary statistics. So we have submitted 855 specimens so far, which is 9 of those plates. We've received 848 sequences. And so if you compare those two numbers, we've only had 7 sequences that completely failed, which is really, really successful, which means that all of the bees everyone's collecting for the Oregon Bee Atlas have super high quality DNA and are super successful using these methods. That includes bees that have come out of our traps, which is really encouraging. So we have 848 sequences. 729 of these are considered to be barcode compliant, which means they have to be a certain minimum length. And then you can see that all of our spatial data and all of our images are fully populated for all of our specimens. Now on this landing page for the project, there's some other summary statistics, and these are mostly project-based statistics about how quickly our project is growing, how good quality or how reliably the DNA samples are able to be sequenced. You can see the sequence length distribution, which shows that the vast majority of our specimens have very long sequences, which is an indicator of high success. It thinks that there's 223 species within our material, which is, I would say, close. But the thing that we really want to see are the records. We want to see the actual bee records within our barcoding project. So here's our records. Each one of those bee legs we submitted that corresponds, to an image and to all of these data sheets is summarized here. And so each row represents one of those bee legs, one of those specimens that's been sequenced. Here we have the taxonomy for the specimen. So these were identified by Joe Engler, that's the JE. These are morphospecies codes because we don't know what species these are. And so this SF stands for Sphacotogastra subgenus female. And then this would be species CDV11. Now, each one of these bee specimens has a specimen page, and that's where a lot of those data sheets ended up. So we see that photo that was taken, this one by Cody Feuerborn. And then we have all of the specimen details, the taxonomy, and some additional information, the collection data, and just the point on the map. So this specimen was collected by Mark Gorman. Thanks, Mark. In 2019 in Hillsborough, right near Portland. And it's a cool Lasioglossum of some kind. So each specimen gets one of these specimen pages. And then once the bee has been, the bee leg has been ground up, the DNA extracted, amplified, and then sequenced, each one of these bees gets a sequence page as well. And I assume you can see these different pages popping up. Okay, thanks. So once the sequence data is put online for us, they provide like this illustrative barcode, which is a colorized representation of the nucleotides. They provide the actual nucleotide sequence, a converted amino acid sequence here, and then the actual trace files where we have the amplitude for the nucleotide at the position. And you get a forward read, and in this case, a reverse read as well. This is amazing. But that's fantastic, you know. Okay, so we got all these bee DNA sequences now. You can see here the sequence length. So a full code would be 658 base pairs. So you see some of the Lasioglossum are below 658, but they're still very informative. But you see that in general, our sequences are very long, and you don't see too many of these where there's an error, a high level of ploidy, so multiple chromosomes and different issues arising. Most of they think these ones are maybe contaminated. But for the most part, we have like high 90% success rate. So once you have all these sequences, oh, this is great, but what can we do with it? And so what I'll do is I'll show you an example. You can either select these records directly from here, just by clicking on the box and toggling the individual records on and off, or you can search your entire project or potentially all public records for any particular taxa you might be interested in. And that could be just Lasioglossum. It could be all of the Halictini tribe. It could be all of the Halictidae family. And that could be for any region, either for the USA, for North America, for the Nearctic, or for the world fauna, whatever sequences might be available to you. So, and once you've selected some sequences you want to analyze, the great part about this bold Barcode of Life data system is that it has all of these analytical tools built in. So what we're going to do is we are going to do a record search, and we're going to find all of the high quality osmia records for CO1 data, sequences that are 600 base pairs long minimum, so all of our really good ones, and just from the project ORB. So this will just be our high quality osmia records. So here now we have our 206 high quality osmia records for our Oregon barcoding project. So now you can see all these different osmia. They have super high quality sequences because I just kind of filtered for that. Just another example of a specimen page. And so what we can do with these is we can select them all, and then we can generate a neighbor joining tree. So like a family tree for all of these mason bees. Now, it takes about a minute or so to do this. I'm not going to do it. I already did it. And so we'll just skip straight to the tree, which is right here. So this is the output. 206 records. Remember, we just saw that. And so what we get is this amazing tree automatically produced by the software. So what we have here is sequences that are grouped based on their similarity. And so the difference between these groups is described by the length of these branches. And this is the divergence in the sequence. So this would be maybe 4%, just comparing it against our bar. And so we could say that this whole group has 4% divergence from whatever this cluster of specimens in one species is. So just to reiterate that, highlight. These first 4 here are all in one species group. And they're very significantly divergent from what looks like 3 additional species here: Californica, Montana, species that we're actually familiar with, maybe, and Subaustralis. And to take it a step further, all of these species in this one branch, or this one cluster, or this one clade, are all in the same subgenus. These are all Helicosmia. So if we continue, we can see other good examples where these Osmia cala are all on the same line because there's almost no difference between those sequences. Those sequences are almost identical. And so they group up and there's no branching in this group. However, those Osmia cala sequences are 3, 4, or 5% divergent from this group of Osmia regulina. And within this group of Osmia regulina, we see a tiny bit of divergence here. So there's a little bit of, maybe 0.2% difference between these sequences among these individuals of the same species. I hope everyone's following that pretty well. I know Kate is, for sure. And so you can see that many species are well resolved. We see that Albolateralis has very little variation within the species and has quite a bit of divergence from its nearest neighbor, an unknown Osmia species here. And so this is all of our Oregon Osmia species. We've actually detected more than 75 species of Osmia in the state so far through the Oregon Bee Atlas, which is, I love it. It's amazing. And so one example I'd like to show you is this Acanthosmioides species group. So historically, there was a subgenus in Osmia, Acanthosmioides. It has been recently sunk into Melanosmia, which it's got like all of the Osmia species in that one subgenus now. But within our results, most of these are all Acanthosmioides group species. And if we look, what we saw earlier, these exigua come out nice. But if we look here with all of these Acanthosmioides, it's a pretty big mess. Right? There's all this variation everywhere, and the names don't match up. Whatever I identified as Physaria, it looks like it is the same thing as Obliqua. And then we find Obliqua way up here by Odontogaster, and so on. And so this group is not well resolved, and that could be for many reasons. We really don't, or at least I don't really know why. So, and then we also see that there's this little outgroup within this clade, these 2 specimens that don't have a name. And so that's great. We can barcode lots and lots of Oregon bees and Oregon mason bees, Osmia, and we can learn a lot. It can help us generate reference material. It can help us associate the males and females. Most of the Osmia I barcoded are actually males, because there's more reliable resources for identifying the females. And so one thing we can do is we can collaborate with other people and make use of public data. And so this is the Osmia results for Oregon. And so here I took all of the publicly available Osmia results, 2,251 records. And we can take a look at that same Acanthosmioides group and see if we can make any more sense of it. So as the data set gets bigger, it gets more highly resolved. And so now you can see that many of these species groups here, they come out really, really well, and it makes things a lot easier. So we are going to go down to, I'm just going to jump down to our Acanthosmioides group and see if we can make any sense of this. So we see that there's some additional species within that former subgenus, Alpestris, Giffardi. So those are ones that we didn't have in our data set, and maybe Paradisica as well, although that doesn't seem like it should be in here. But we see that they also, the Alpestris come out together, Giffardi come out together, Lanei, or whatever I call Lanei, comes out independently. We see Sladeni and Nithoata now together. We see a long group of Odontogastr. And so maybe it's more resolved than it was, maybe not. It's hard to say. But this can inform us in many different ways. And then finally, you'll recall we had those two bees that had no names. That was this outgroup. And so sure enough, here we find that same outgroup with our two bees that are lacking names. And so, what are these? We also see that someone else probably has a specimen of the same species. So all of our sequenced bees have all these different kind of identifying codes on them. So that these two bees are 330 and 334. So we can actually just hop back and find them and find the photo and see where those bees live to see maybe we can do some sleuthing. So you recall 330. So let's just take a look at the specimen pages for those two specimens with no names. And so these are male bees, definitely in the Acanthosmioides group. Acanthosmioides males have all these spines on their bellies. They have a super dense mustache, and the tips of their antennae are often flattened and rounded. And this bee was caught by Julie Biddle way out, it looks like in Harney County. So this is out by the Steens somewhere, maybe. Yeah, it looks like somewhere out there. It's hard to say. And then the second specimen was 334. So we can take a look at this one. It's another male. It's another Acanthosmioides species of some kind. And this bee was caught by Tom Robinson, Julie's husband, in the same region, certainly on the same field trip. And so that's really good. We have this like unknown species in a known species group. We have some good genetic data. It matches up with someone else's genetic data. Maybe their specimen is a female. So then we could have the associated male and female, and we can see if they match up with a known species in that species group. And so you can see how informative this, this data could potentially be. So we're going to move on from that. And you get a sense of how this works, what's available in the Barcode of Life data system. And the neighbor joining tree is a tool that we use frequently. It's a good way to visualize the relationships between these things. But you can pump out tables that literally will tell you the percent divergence between and within all these species and their neighboring groups. And there's a dozen other tools as well, which I won't go into, but get yourself a login for the site and you can make a data set and do all these kinds of things yourself. So over the last, I would say, well, during the pandemic, Joe Engler spent a lot of time working on the Oregon Bee Atlas Lasioglossum specimens, as well as material that he had collected from Pacific Northwest refuges while he was working with the U.S. Fish and Wildlife Service. He has identified at least, I think, 8,800 Lasioglossum specimens for us. So it's St. Joe. And we have DNA barcoded at least 342 of those specimens. He's been working on a really great manuscript. And so here are some of those results. We've focused our barcoding effort on the, let's see, small, non-metallic Lasioglossum subgenera. So really focusing on the Sphicotogastra, the Hemihalictus, and the Evolaeus, everyone's favorites, and to a much lesser extent, the Dialictus, and Leucalictus and Lasioglossum sensu stricto are not even not represented here at all. And mostly they're well known, the two subgenera, so we don't really need to barcode them in many cases. But even in here, this is just a tree and a diagram I put together for this talk. We can see that this group here is the Sphicotogastra. And you can see the long branches again, which I've highlighted in red, which are the differences between these species. So here we get one species group, two, just represented by two specimens, a third, a fourth, a fifth, a sixth, and a seventh. And then we have the Dialictus group here, which has 21 branches. We have the Hemihalictus group here, which has 9, and then Evolaeus, which only has 3. Joe has done an incredible job resolving a lot of the taxonomic issues within these subgenera, especially Evolaeus, Hemihalictus, and Sphicotogastra within Oregon and beyond. And so we're hoping to get a manuscript out in the next few months anyways. And we'll release a lot of this barcode data along with the manuscript so that other people can match their bees up with our bees. And so even if you can't necessarily identify it under the microscope, because some of them are still difficult or impossible, for $20, you could potentially sequence your bee and then just match it up to our code. And it'll tell you for your project that morphospecies has a name, or maybe it's an undescribed species because Joe has identified a handful of new species within some of these groups, and so on. Just a really powerful tool. So of course, all of these specimens have come from our Oregon Bee Atlas volunteers. It's really amazing the work people have put in. We're almost at 20,000 samples. These are just some of the dots of the Lasioglossum we've sequenced to give you a sense of the coverage. But it's just a lot, and it's amazing. And so following up on that, we have been successful this spring in a grant through U.S. Fish and Wildlife Service and WAFWA, the Western Association of Fish and Wildlife Agencies, through bipartisan infrastructure funding to the Department of the Interior for sagebrush biome conservation. And so we're successful in a grant that will produce bumblebee data for the high desert in Oregon, which is sagebrush biome, as well as funding for the production of DNA barcodes for solitary bees in those same habitats within the sagebrush. And so yesterday I received 20 more plates from the Canadian Center for DNA Barcoding. And so we hope to contribute about 2,500 more specimens and generate that many more sequences beyond the 855 that we have already. We already have more than 200 species sequenced, and we expect that this will get us hopefully over 600, a reference collection of sequenced 600 plus species would be just an incredible resource for everyone in the region. So once again, thank you to everyone at the Oregon Bee Atlas for all your incredible contributions. You know, all of these specimens, these 2,500 specimens we're going to sequence, it all comes from everyone that's out there taking pictures of plants, catching your bees, going home, pinning and labeling your bees. So all of that hard work is growing into this incredible public resource. So with that, I'll introduce you to what I originally called the FUS, and this is controversy now coming out of DNA barcoding. So one of the implications of being able to sequence hundreds, thousands, tens of thousands, or even millions of individual organisms and compile these massive genetic databases is that, well, within some groups, even some groups of bees, we have many new species. And it is, if you were to revise one genus, it could take you a master's, a PhD, and a postdoc. It might be a 12-year mission to revise one genus, and maybe not for the world, maybe just for North America, you're revising that genus, or longer in the case of some genera that are super speciose, like Lasioglossum. And so what's possible is that we could describe those new species potentially just with the genetic data and an image. And that's what Michael Sharkey and colleagues did here for more than 400 new species of parasitoid wasps, mostly reared from hosts in Costa Rica. And this caused quite the uproar, because instead of doing all of the background revisionary work to find out what's a synonym and produce these really long descriptions that describe every angle of the mid-femur, they're just publishing a sequence, which is diagnostic potentially, and an image to give you a sense whether you're close or not, really. And so, the consequence of this was a lot of criticism within the scientific community. As you might imagine, as things change, there's going to be a lot of discussion about that. And so this is, the discussions about this are quite fascinating. Should we do it? How should we do it? Is it reasonable? Is it out of control? Is it manageable? Is it not? Should we use the bins that the Barcode of Life data system constructs using algorithms that aren't fully clear to everyone, and which are certainly not always representative of actual species concepts? And of course, Sharkey et al. Responded to their critics, and it just goes back and forth. And so we can have a discussion after this about whether that's reasonable, but I have to show you, of course, what they did. And so this was their species description, and they had more than 400 of these. The diagnostics, rather than a description of the morphology, which is the traditional method, you would describe what the organism looks like and image the genitalia, and so on. They just provided this barcode sequence, like you just saw that we have for our bees. They provided the specimen details, that it's going to be deposited at the Canadian National Collection. Some of the, specimen data, identifiers, the basic stuff. And that's about it. So we got the code and we got the photo. And the big question is that enough for a species description? Because even in Oregon, even with the bees, we have this huge challenge that within some of our genera, we may have dozens of new species. Our results for Stelis are amazing. When we barcode our nomata, we're going to find like 50 species in Oregon, and we're going to be able to put names on 5. And so should we do like what you saw with Joe's work and apply a morphospecies to it and call it a number and with a prefix, or should we just give them names now that we have a diagnostic barcode for them and an image? Because realistically, it might be 20 years before we get a key to the nomata of any part of the Western US. Maybe there'll never be one. And so maybe we should do this and at least have a name that we can work with rather than having a morphospecies name, which is basically the same thing anyways. So it's an ongoing debate. And with that, we can continue the debate, but I'd like to thank Jerry and Judith Paul, Ray Temple and Stephanie Hazen, Missy Martin, Jean Natter, Pam Hayes for their generous donations, all of our Oregon Bee Alice contributors, many of you in Washington that can't help yourselves but come down here and contribute. And of course, all of these funding groups on the right, who we would not be able to do this without. So thank you very much.